Okay, everyone, it's 2:30. It's time to begin. I am so thrilled to see so many members turning out and guests to celebrate this year's awardees at the National Academy. It's just really great to see you all here, and I want to give a special welcome to those who are joining us online to find out all the amazing things that scientists have done in the past year. I want to let you know that the cycle for nominations is going to start all over again in October. And one of the struggles that the selection committees have every year is getting a wide enough range of nominees that they can actually make, interesting and impactful decisions. So if you go to the NAS website, there will be directions on how to nominate someone for one of these awards, and I very much encourage you to do that. I'll also say about our awards, we are working very hard to give them to earlier career scientists because of the impact that an award has on the trajectory of a young person. So think of young people to nominate as well. Okay, first up in our award ceremony are the Cosarelli Prizes. The Cosarelli Prizes are named in honor of the late PNAS editor, Nicholas Cosarelli. The prizes are awarded annually for 6 different research teams who published their work in PNAS. The 6 teams represent the 6 classes in the Academy. So you'll see as we go through this, one for each class. First up is Class 1, Physical and Mathematical Sciences. And the Cosarelli Prize is being awarded to the paper Permafrost Thaw Subsidence, Sea Level Rise, and Erosion are Transforming Alaska's Arctic Coastal Zone. Coastal erosion is a growing hazard along Alaska's Arctic coastal plain. Erosion is compounded by sea level rise, land subsidence from permafrost and thaw, and also from the retreating ice cover that used to protect the shoreline. The authors of this study found that without mitigating measures, land loss could damage 40 to 65% of infrastructure in villages on the Arctic coastal plain, and 10 to 20% of oil field structures in the Alaska North. Class number 2 is Biological Sciences, and the Cosarelli Prize is being given for the paper Eye Blinks in a Visual Processing Stage. I found this one amazing. Blinking mainly serves to lubricate the eyes. That's what we've all also thought. But blinks also frequently interrupt the visual information that is coming to your brain. The authors of this study found that blinking increases the contrast of different visual stimuli and improves the visibility of low resolution information that's in your visual scene. So rather than impairing vision, blinking actually facilitates visual processing by enhancing the sensitivity of what you perceive. For class number 3, which is Engineering and Applied Science, the top paper was Distribution of Centrality Measures on Unidirected Random Networks via the Cavity Method. So complex networks are often used to represent so many things around us. We have social networks, we have technology networks, we have biological networks, and of course we have financial networks as well. A key challenge in analyzing complex networks is quantifying centrality. And what's centrality? Centrality is a measure of the importance of each of the nodes in that network. And the authors of this study provided an algorithmic method to identify the key nodes for the functionality of the real-world networks, such as global transformation networks. So they found those linchpins that if you pull those out, things collapse. So that's where you have to invest first to protect them. Next up is Class 4, Biomedical Sciences. And the award is being given for ultra-high frequencies of peripherally matured GLI1 and Casper-2 reactive B cells characterize the cerebrospinal fluid in autoimmune encephalitis. So autoimmune encephalitis is characterized by polygenic antibodies that react to your central nervous section proteins, often resulting in cognitive impairment and relapse despite treatment for encephalitis. The authors of this study characterized the pathogenic immune cell receptors from the cerebrospinal fluid of patients targeting 2 different proteins. Their findings could aid in the development of targeted therapies for autoimmune encephalitis. And then for Class 5, the award, which is Behavioral and Social Sciences, it's for the paper AI Accelerated Nazca Survey nearly doubles the number of known figurative glyphs and sheds light on their purpose. So for over a century of work in the field, very hard work, researchers have found 430 Nazca geoglyphs on the southern coast of Peru. So these are early American carvings and drawings on the south coast of Peru. The authors of this study used AI to analyze aerial images of the region and predicted and then confirmed the presence of 303 additional glyphs that had never been found. And they did this in seconds compared to the century to find the first 430. The analysis revealed both relief-type geoliths, like you have here, and line-type geoglyphs featuring wild animals. And for, last but not least, for Class 6, Applied Biological, Agricultural, and Environmental Sciences, the award is given for the paper Prey Can Detect Predators via Electroreception in the Air. And I found this fascinating too. Predators and prey detect each other for a variety of senses, our eyes, ears, nose, whatever. But one such sense that prey can use to detect predators is electrostatic charge. The authors of this study report that predatory common wasps carry an electrostatic charge and that prey can use electroreception to detect these predators that are coming for them. This phenomenon may be more widespread among terrestrial animals than we currently know. So I want to say congratulations to all of the authors on these 6 outstanding papers, and let's give them a round of applause. Okay, and now on to our individual awards. The first up is the Alexander Hollander Award in Biophysics, and it's awarded to Louis E. Kay of the University of Toronto. And it's for advancing the field of NMR spectroscopy by developing techniques to study biomolecular dynamics in rare conformational states, extending the use of NMR to large systems, and contributing to our understanding of molecular machines, protein folding and misfolding, with significant biomedical and therapeutic impacts. So I would like to invite Dr. Kay to give his acceptance. We hope you accept. I hope you do.
Well, thank you very much. I'd like to begin by saying how grateful I am to be the recipient of this award in biophysics. I want to thank the National Academy of Sciences. I want to thank the Hollander family and the selection committee for their confidence in me. It's always nice to get an honor and, most importantly, a pat on the back and told that your work is meritorious. And in that regard, a special honor for me to be here today. But the real honor for me is less about winning this honor and just having the pleasure of being back in Washington, D.C. You see, many years ago now, I was a postdoctoral fellow at the NIH in the Laboratory of Chemical Physics in the sub-basement of Building 2. Now, you have to understand that in those days, logic was pervasive, and so I'm going to assume that Building 2 was one of the first buildings that was built on NIH campus, perhaps even the second. And it really showed its age. But what was absolutely amazing is what happened inside the building. The science was absolutely fantastic. I got to work with some of the world's brightest minds. And in that regard, I really want to acknowledge Ad Bax, my mentor, who's here today, for his help and guidance in making me into the scientist that I am today. I also want to acknowledge Dennis Torsha, who was a very important mentor figure in my scientific development as well. I'd like to acknowledge Jim Prestegard, another mentor, a graduate supervisor at Yale University, and then my undergraduate supervisors, both Brian Sykes and Ted McClung at the University of Alberta. Now, it's also an honor for me to be here today to say thank you to the National Academy of Sciences. 5 years ago, I was elected an international member, but could not attend because of the COVID situation. So it's particularly gratifying to be getting a second opportunity, one where I can come in person and say thank you for this honor. And I also want to thank the National Academy of Sciences for really championing the critical role of science, not only in the US, but also on the world stage in general. Those of us who come from abroad look up to the NAS for guidance and support during these difficult times. Finally, I want to thank my trainees over the past 30-plus years who have enriched my scientific career, my parents and sister for serving as role models, my wife Julie, who has been my backbone. She joined me at the NIH for the completion of her PhD and then for postdoctoral training. And while we have collaborated occasionally on a variety of different scientific projects, no doubt that our two most important collaborations are Ruffy and Shira, our two children, and I'm delighted that they're here with us today as well. I would also like to thank Aunt Sherry for joining us in today's celebrations. My family has taught me that there are things more important than spin-angular momentum, or than finding that elusive minus sign in one of my equations, which is driving me crazy, or even then elucidating the conformational landscapes of proteins. My family continues to be, for me, the biggest prize of them all. Thank you so very much.
Our next award is the Arctosky Award, and it's awarded to Stuart D. Bale of the University of California, Berkeley. It's for his seminal work in developing instrumentation, particularly for the Parker Solar Probe, and for interpreting the resulting observations that have revolutionized our understanding of the energization of and heat transport in the solar wind. So please, Dr. Bale.
Thank you, Dr. McNutt. Thank you to the National Academy of Sciences, and thank you to the Arctosky family for this award. I'm deeply honored to receive the 2025 Arctosky Medal. The recognition reflects not just my own work, but also the work of many collaborators and students and engineers and my mentors as well. And of course, the important support of NASA's Science Mission Directorate. So the medal, I consider this medal really to be a tribute to decades of collective work to understand our sun and the plasma astrophysics associated with it. And my role is just a small part of that. Thank you.
And now we have the Henry Draper Medal, which is being awarded to Adam K. Leroy of Ohio State University. And this is for his path-breaking efforts to characterize in unprecedented detail the physical nature of the interstellar medium and its relationship to star formation in nearby galaxies, and for his visionary leadership in building the next generation of multi-wavelength, high-resolution surveys and creating data products for the community of extraordinary legacy value. So please, Dr. Leroy, come join us.
Thanks. So I'd, of course, like to say thank you, and this is extremely humbling to the National Academy. I also think this particular prize is extremely neat for someone who studies the interstellar medium, since among many other accomplishments, Henry Draper took the first photographs of the Orion Nebula, which is super cool if you study nebulae. So I'd like to acknowledge three things here. The first is that none of the work that's being acknowledged here would be possible without an amazing network of international collaborators, including Ava Schinnerer, Eric Rozalowski, Karen Sandstrom, Alberto Bellato, and Chai Sun, as well as the entire amazing FANGS team. Very important, I think especially at the moment, is also that none of this work would be possible without the incredible US national observatories, and in particular, the National Radio Astronomy Observatory run by NSF has been crucial to the work that we've done, and the many great observatories run by NASA are absolutely essential. These things are really wonders of the world, and we should be working as hard as we can to improve and preserve them for the next generations of scientists. Finally, I'd like to acknowledge the amazing scientific atmosphere at Ohio State, and last but absolutely not least, my amazing family, including my wife Jessica, who flew in to be here today. Thank you very much.
Thank you. And now we have the James Prize in Science and Technology Integration. This year it's being awarded to Jennifer A. Lewis of Harvard University for integrating multidisciplinary expertise in material science, soft matter physics, additive manufacturing, bioengineering, and developmental biology to create printable inks and multi-material 3D printing networks for manufacturing materials with a wide range of physical properties, enabling technological applications ranging from electronics to biomanufacturing of vascularized human tissues. So Jennifer, please join us.
Thank you so much, Dr. McNutt, to the NAS, and also to Bob James and his family for their generous support. I am deeply honored to share this recognition with my students and postdocs who have made profound contributions to our research and also have had a profound impact on my life. I'm also grateful to the University of Illinois and Harvard University for providing an exceptional intellectual home for our research over the past 3+ decades. And to our many collaborators, we certainly couldn't have had an impact on even a small fraction of the fields that you just mentioned without the mentorship and the scientific discussions and stimulation that I've had with many colleagues, and I want to name a few. Ken Schweitzer and David Waits and Chip Zukowski in soft matter physics, Joanna Eisenberg, Katya Bertoldi, Paul Braun, Ralph Nizzo and Nancy Sotos in material science, Joseph Cesarano in 3D printing, Howard Stone in fluid mechanics, Dave Mooney and Don Ingbar in bioengineering, and in our work in trying to advance the frontiers of kidney tissue engineering, I want to thank Annie Moisen, Leo Raella, a clinical collaborator at MGH, and Lisa Satlin, another clinical collaborator at Mount Sinai. And lastly, I want to thank the program managers that have been in the trenches every day working at DOD, DOE, NIH, and NSF, and as well welcome LEAP for their generous support of our research, without which none of this would have been possible. So thank you so much.
Okay, next up is the Michael and Sheila Held Prize. This prize has been given to a team. So the team members are Nina Anari of Stanford, Kwei Liu of the Massachusetts Institute of Technology, and Shayan Oves Garan and Cindy Vincent, the last two both from University of Washington. So please, team, come up. Oh, I'm sorry. Wait. I forgot to say why you're getting the award, what you did. All right, let me say what they did. They bridged the theory of high-dimensional expanders, the geometry of polynomials, and the analysis of Markov chains to resolve the Mikhail Vazirani, is that right? Conjecture that is the basis exchange walk on a matroid mixes wrap on a matroid. I'm sorry, let me try this again. This conjecture that the basis exchange walk on a matroid mixers rapidly and for initiating the influence of theory at spectral independence. Maybe they'll tell me what I just read.
All right. Let me first thank the Health family and the National Academy of Sciences and the selection committee for this award. I also like to thank God for all the opportunities I've had in life to focus on right problems with right tools and collaborate with creative scientists. I also owe this to my family, Farnaz and Faraz, and my parents for their love, support, and bearing with me in difficult times. In addition, I'd like to thank my advisors, my collaborators, and my students over years for shaping and helping my research and my career. Now, a little bit about our work. Matroids are abstract objects which generalize linearly independent set of vectors in a vector space. Their geometry, combinatorics, and algebra has puzzled researchers for years. Our work shows that one can sample a uniformly random base of a matroid using a Markov chain efficiently. For example, it shows that, it gives an efficient algorithm to sample a uniformly random base of a given set of vectors. Since invention, our method has found many applications across science, in math, physics, and computer science. In particular, it has led to efficient algorithms for sampling from the hardcore model of gas molecules in a statistical physics.
Thank you. Thank you. Now I understand perfectly.
So Cheyenne said what our work is about, so I'll just constrain myself to thanking. So thank you to the nominators and the committee and the National Academy of Sciences. I'm incredibly honored to receive the Michael and Sheila Hill Prize. I, of course, owe a great deal of gratitude to my mentors and collaborators, in particular Cheyenne Kuikui and Cynthia, who couldn't be here. They showed me the value of patience and focus in research. They also empirically proved to me that very good things can come out of going out of your comfort zone and talking to people who are outside of your immediate area. Our work, in fact, started as a conversation between a group of people who were interested in seemingly distant areas, algebraic geometry, algorithms, and probability. I would also like to thank the Simons Institute for the Theory of Computing, who actually hosted a group of us and brought us together, and that was the start of this unplanned collaboration. I would also like to thank my family, my wife, Dorsa, who is at home watching, together with our 10-month-old son. All right, thanks.
All right, yeah, so since Cheyenne already talked about the science, I'll just give my thanks. So I'd like to thank, first of all, the National Academy of Sciences, the Health family, as well as the selection committee for this award, which I feel extremely honored. I'm immensely grateful to my PhD advisor, Cheyenne, as well as my wonderful collaborators, Nima Anari and Cynthia Bizant, who couldn't be here today, as well as the Theory of Computation group at the University of Washington, where I completed my PhD. I would also like to thank my family for their unwavering support, in particular my partner, Clara Liu, my parents, Junyi Liu and Wei Xu, my sister, Lily Liu, and my wife's parents, Wenxin Lu and Yinghong Du. Thank you, everyone.
Now, as you heard, Cynthia Vincent couldn't be here to accept the award, but she was kind enough to actually record some remarks to show here. So go ahead and play Cynthia's.
So I'm sorry to not be able to join you all in person. Firstly, I'd like to thank the National Academy of Sciences, the prize selection committee, and our nominators for this honor. I'd also like to thank the Simons Institute in Berkeley, where I first became involved in the work being recognized. I'm very grateful for and conscious of the openness and generosity I've experienced in the mathematics and computer science communities throughout my career, in particular from my collaborators and my academic mentors. It's been a joy and an honor to get to work with people with such intellectual curiosity. Lastly, I'd like to thank my partner and family for all of the support and encouragement over the years, without which none of this would have been possible. Thank you.
Okay, next award is the NAS Award for Chemistry in Service to Society. It's being awarded to Michael E. Jung of the University of California, Los Angeles. It's for his design, synthesis, and development of enzalutamide, otherwise known as Xtandi, or apalutamide, otherwise known as Erleada, the most successful drugs for the treatment of both castration-resistant and castration-sensitive prostate cancer, which have extended the lives of literally thousands of men worldwide. So please, Dr. Yang.
Thank you very much, Marcia. I want to thank Pete Schultz, who nominated me for this award, the selection committee for choosing me for this great honor, and the NAS for recognizing that chemistry can be a good thing, not just a bad thing. Chemists do a lot for the world, and we've tried to help a little bit in terms of biological things. I have to thank my wife, Alice, who convinced me when I was 55 years old to change my research focus. I was an organic chemist and then became a medicinal chemist, a drug discoverer. And it's a new world, and it's exciting, but it's scary when you're 55 to start something brand new. But I did, and it seems to have worked, so. I want to thank the two students who did all of the work. I claim the prize, but they did the work. So Samadi Ook and Dongwon Yu, both postdocs in my lab. Samadi did the lion's share of the work, and Dongwon finished it off. I want to thank the biological team of Charles Sawyers and his folks who did all of the biology. I don't know any biology, but they did all the biology that led to the discovery of these two drugs. And I want to thank the rest of my research group at UCLA and my colleagues there who've made a hell of a good environment for doing drug discovery. This is my 51st year at UCLA, and I'm still hanging in there for a while. In fact, I asked Jenny Lee if I could say a little bit about science while I was up here. So I'm 78 years old, and I haven't retired and have no plans to until I get another drug on the market. And we'll see whether we can. I have 4 drugs that are in the clinic now, a DCK inhibitor for solid tumors and multiple sclerosis, a compound that causes mucosal healing for Crohn's disease, inflammatory bowel syndrome, and ulcerative colitis, a brain-penetrant EGFR inhibitor for glioblastoma, and then one that has no therapeutic value at all. We have a small molecule, when applied to the skin, causes hair to grow. And I can look out and see that there's several people that could be using this. It's in clinical trials. It's in phase 2, the open label extension. We're about halfway through phase 2. And if this works, that may be another example of the power of chemistry to help certain parts of society. Once again, I want to thank the NAS very much for this award. Thank you.
Well, my money is certainly on Michael to bring these drugs through, but not necessarily on the FDA for quick approval. So we'll see how that works out. Okay, next up is the NAS Award for Scientific Discovery. It's being given to Xiaodan Xu of the University of Washington for the experimental observation of the fractional quantum anomalous Hall effect in topological materials, revealing fractionalized electronic charge carriers without application of a magnetic field.
I would like to thank the nominator, the Academy, and also the selection committee for this incredible honor. Our discovery builds on years' progress of essential community, such as quantum Hall effect, 2D materials, topologies, interactions, magnetisms, making this award a celebration of the entire community. So I want to thank my students for their hard work, especially to 3 brilliant students, Eric Anderson, Jiaqi Cai, and June Park. I also want to thank my wonderful collaborators, especially to Wang Yao and Di Xiao. They taught me so much. And I'm grateful to my family, my wife, Ying, my boy, Tyler, and also my parents back in China. So my mom, actually, it's a typical Chinese mom, tiger mom. So every time when I see her, I imagine I took more than a 10-hour flight. The first thing she asked me is, have you done anything significant? And I told her that, Mom, I'm not a kid anymore. Hopefully next time when I see her, I share this award with her, make her proud. So finally, I'm thrilled about the potential of our discovery, and I think this is just the beginning of an exciting journey in science and technology. Thank you all.
Now we have the NAS Award in Chemical Sciences. It's being awarded to Peter G. Woolness of Rice University for far-reaching contributions to chemistry, illuminating mysteries that range from quantum chaos in molecules to the nature of the glassy state, and for resolving paradoxes of protein folding with the energy landscape theory and its associated tools that predict protein structure and functional dynamics from the sequence. Peter.
Thank you, Marcia. First of all, thanks to the Academy and to the selection committee. It's a great honor, and I've been honored by the Academy by being a member for quite a while, and it's always nice to know that people think you're still doing okay after a while. My area of science is physical chemistry, which was defined by G.N. Lewis as the study of anything that's interesting in science, and I think that's why it's been a wonderful area for me to be able to work on. I've had, therefore, wonderful collaborators, wonderful students, and I think one thing that this award brought to mind is that in every case, simply making a discovery or an inroad on a problem isn't enough. One has to build a whole community, and in the more successful areas of my work, that community has been built, not by me, but by the members of the community, and has really resolved questions that I really wanted to know the answer to. So I appreciate everyone in the community of physical chemistry and biophysics who've helped with this. And of course, I want to be sure to thank my wife, Kathy, who's heard the grumpy version of this talk about the community and has always kept me, on the straight and narrow. I haven't always followed her advice. I haven't fired all the graduate students she told me I should fire. But I would have done better if I had. And I thank her and my daughters, two of whom are here, and I appreciate their support. Thank you.
Okay, now we have the NAS Award in Early Earth and Life Studies, otherwise known as the Stanley Miller Medal. It's being awarded to John D. Sutherland of the MRC Laboratory of Molecular Biology for his pioneering work on prebiotic chemical pathways and geochemical scenarios that together provide fresh insights into the synthesis of the building blocks of biology in the early Earth. Now, unfortunately, John couldn't be with us today, but he also has sent an acceptance video, which we can view now.
Good afternoon, everybody. Welcome from England. I'd just like to say thank you very much on behalf of myself and my research group for this award, which is really a great honor, and I really appreciate getting it from my friends and colleagues in the US.
Okay. The usual British crispness. Okay, now we have the NAS Award for Molecular Biology. And that's being awarded to Eric Scarr of Vanderbilt University Medical Center for his pioneering discoveries in nutritional immunity, uncovering microbial strategies to acquire metal ions during infection and immune counter strategies to mobilize metals to prevent infection. So, please, Eric.
Thank you for that kind introduction. This award is an incredible honor for me and a highlight of my career. I'm very thankful to the National Academy of Sciences for selecting me for this award, and to my colleagues who nominated me for the award and provided letters of support. There's no way that I would be here today if it wasn't for the incredible support and mentorship that I received from my undergraduate advisor, Dr. Timothy Donahue, my graduate advisor, Dr. Hank Seifert, and my postdoctoral advisor, the late Olaf Schneewind, who was a member of this academy. The discoveries that our lab has made at the intersection of infection and nutrition are the result of an incredible amount of hard work and dedication from a number of very talented people. And I just find it to be an incredible privilege to spend each day with these people who have devoted themselves to a career in research, who will be the future leaders of research and are so focused on discovery. They inspire me every day, and I'm very thankful to them. And then finally, I just want to thank my wife, Heather, and my daughters, Hannah and Ella, who are incredibly supportive of me, and they have always been and will always be my primary motivation. So thanks again to all of them, and thanks to you.
Okay, now we have the NAS Award in the Neurosciences. It's being given to Li-Ching Hu of Stanford University for his contributions to the development of genetic tools for neurocircuit analysis and his work uncovering foundational mechanisms of neural development, neuronal diversity, and brain wiring. His seminal work spans molecular genetics to systems neuroscience, and his widely acclaimed textbook has become a cornerstone of neuroscience education worldwide.
Thank you very much, Marsha. And I would like to thank the National Academy of Sciences, the nominators, and my neuroscience colleagues on the selection committee for this great honor. I'm extremely grateful to the graduate student postdocs in my lab for the last 28 years at Stanford, whose work, whose dedications and talent has contributed to all the work that's being cited. I'd like to give a shout out to using simpler organisms like fruit flies to uncover general principles of neuronal development and function that can apply to complex brains like ourselves. And since the award also mentioned my textbook, I'd like to thank numerous colleagues and students who give advice and feedback. And particularly, I'd like to thank the Rupert Stryer, whose advice and encouragement has been instrumental for my having the textbook, and his lifetime pursuit of the relationship between light and life has always been an inspiration. Thank you.
Next up is the NAS Prize in Food and Agricultural Sciences. And this year it is awarded to Hans Kotze of Kansas State University for his transformative research into safe and cost-effective pain relief solutions for farm animals, which has led to improved health outcomes and reduced antibiotic use.
Thank you, Dr. McNutt, and thank you to the National Academy of Sciences for this incredible honor. I also appreciate the Gates Foundation and the Foundation for Food and Agricultural Research for endowing this award, and to the selection committee and the nominators for preparing the letters. And in particular, I'm appreciative for the awareness that this has raised of the challenges and opportunities farmers face in easing livestock pain. As a child, I watched Holly, my show heifer, be dehorned without pain relief, which inspired me to become a veterinarian focused on animal welfare. Annually in the United States, 130 million piglets and 16 million calves experience painful procedures without approved analgesics. Our contribution to securing FDA approval for the first pain medication for cattle lameness represents a first and significant milestone towards improving the welfare and productivity of farmed animals. I'm tremendously blessed to have the opportunity to do this research, and I'm indebted to my mentors, collaborators, students, supporting organizations, and funding agencies for making this possible. We look forward to a future where approved pain relief drugs redefine the standard of care for farmed animals. And to my family, thank you. Your sacrifices and unwavering love made this possible. I'm tremendously blessed to have my wife Tiffany here today and my girls Scott and Annabelle in Kansas watching. Girls, I appreciate all of your support for Dad. I'm standing on the shoulders of giants. Thank you.
Okay, the Pradle Research Award is awarded to Ann Churchland of the University of California, Los Angeles. It's for her pioneering experimental and theoretical research leading to seminal contributions to understanding the neural computations underlying sensory-guided behavior and decision-making. Also for exceptional leadership in neuroscience and promotion of diversity, championship of large-scale collaborative research, and advancement of open science initiatives. So please, Anna.
Thank you so much. I'm very happy to accept this award, both on behalf of myself and also the amazing students and postdocs, first at Cold Spring Harbor and then at UCLA, where I've had my scientific career. I'm also very grateful to the selection committee. I feel very fortunate this weekend to be surrounded by many scientists who've played a key role in shaping my career, including my PhD advisor, Steve Lisberger, my postdoc advisor, Mike Shadlin, and then a number of other people who have really provided excellent guidance throughout my career, including Limor Joshua Tor, Tony Mobshin, John Monsell, and also Katrine Duloc, who nominated me for this award. I'm also incredibly grateful to my family, including my parents, Pat and Paul Churchland, my husband, Alex Huck, and our big blended family of 4 children, including my daughter, Alexandra, who's here today. And I'm very grateful to my family to have guided me both in my scientific career and also my medical journey. I'm a cancer survivor, and I would not be standing here today if it weren't for the amazing support of all of those people. So thank you all very much.
The Richard Lounsbury Award is awarded to Michael M. Yartsev of the University of California, Berkeley, for his seminal contributions to understanding the neural basis of spatial navigation and communication in natural social contexts, and for developing new research paradigms and technologies to decipher neural circuit computation underlying these functions in the mammalian cortex. So, please, Michael.
Wow, this is such a huge honor. Okay, I just want to acknowledge because its contribution is very important. Full disclosure, my daughter helped me write some of those remarks, so just for you to be aware. But I'm tremendously honored, of course, to receive the Lounsbury Award. I'm grateful to the National Academy of Science, the French Academy of Science, also part of this, the selection committee, and of course the colleagues who nominated me and wrote the letters. First and foremost, I want to begin by thanking my family, both near and far. My parents are back in Israel. I know they're watching, even though it's very late. I love you, Mom and Dad. Thank you very much for all your support. I want to thank my wonderful wife, Lisa, without which none of this would be possible. Kind of the scientific journey has taken us on crazy paths, and I don't plan for it to become less crazy in the next few years, and she has really been the cornerstone. And to my wonderful kids, Leah and Ariel, who are truly my greatest inspiration, and unicorns are awesome. I also owe much of my kind of work to past mentors, my undergrad mentor, Ofer Donchin, all the way through Carlos Brody, who shaped my postdoctoral path. And this work could not be done without colleagues in the field who are always generous, some of which are in this room today also, who are always generous with their time, kindness, and advice whenever needed. And people like Edward Moser, Gilles Oran, Bob Knight, Daniela Kafter, and many others I don't have the privilege or the time to thank today. And last but not least, just the people in my lab. They are the ones doing all of this work. You know, they're the ones doing the research day in and day out. We go for crazy questions, crazy projects, which we often think are bound to fail. And it's just their curiosity, the desire and drive that pushes us forward. So I think this whole award is really a celebration of their work and all of their success. And Mommy is awesome. Thank you very much.
So now we have the Selma A. Waxman Award in Microbiology, and it's being awarded to Rotem Sorik of the Weizmann Institute of Science. Sorik's discoveries of immune systems in bacteria have dramatically expanded our knowledge of the molecular arsenal bacteria carry to defend themselves against viral attack. His work has revealed unexpected evolutionary connections between bacterial and eukaryotic immunity and has explained the evolution of components of the human innate immune system. So,
thank you very much. I want to thank the NAS and the members of the prize committee for selecting me for this prestigious award. I'm deeply honored to be included in the same list as the giant in microbiology who previously received this award. Really honored. So the profession of being a scientist is exceptionally gratifying. Society gives us license to follow our passion and have fun exploring the mysteries of nature. I'm in debt to the Weizmann Institute of Science for providing me with the best imaginable conditions to do exploratory science. I want to thank the past and present members of my lab and colleagues whose ideas, work, and passion for discoveries have made this award possible. Special thanks to my postdoc mentor, Eddie Rubin, for helping me shape my identity as a scientist. I want to thank my best friend Zohar, who also happens to be my wife, for sharing the path of life with me and for creating a wonderful family together. And also to my mom, who taught me from a very young age how to admire the beauty of nature, a thing I keep doing to this date. Studying the secrets of microbes has been an exceptionally rewarding journey for me. And I appreciate the recognition today, but this journey continues, and I'm really looking forward to more exciting discoveries in the years ahead. Thank you very much.
okay, now we have 2 Troland Research Awards. The first Troland Award is awarded to Evelina Fedorenko of Massachusetts Institute of Technology for her contributions to the understanding of language networks in the human brain, obtained by combining precision measures of an individual's brain organization with innovative computational modeling. So, Evelina.
Thank you so much. Thank you, the Academy and my nominators and letter writers, especially Randy Buckner. I feel many emotions today. I feel sad because the people who have shaped me, my grandparents, but also my parents, are no longer with us. A special note on my mom, Svetlana, who made me fall in love with math and language, and who taught me to dream really big with no boundaries, which was quite a feat given the extreme poverty that we lived in and the adversities we faced. I'm immensely grateful to my mentors. I learned most of what I know about language from Ted Gibson, who is here today. I liked learning from him, so I convinced him to marry me. It was not easy. And for over 20 years now, he has been my best friend and my biggest cheerleader. And a huge thank you for Nancy Kamwisher, my academic mama, who taught me how to use neuroscience tools in the most rigorous ways to ask the biggest questions about the human mind. I'm also very thankful for my family, including our daughter Lana, my friends and colleagues, and very importantly to all my mentees over the last decade and a half, who make me feel proud every day, but also who give me a lot of hope and optimism for the future and make me feel like it's going to be okay in spite of everything that's happening in the world. So thank you for this recognition.
And the second Troland Award is awarded to Nicholas Turk Brown of Yale University for his pioneering work on the role of attention in visual statistical learning and how this interacts with implicit and explicit memory. for exquisite studies of the neural mechanisms of learning and memory in both cortical and hippocampi circuits using computational functional brain imaging in adults and infants. So please, Nicholas.
Thank you. I'm really honored and humbled to be here with you all today and with many colleagues and mentors. I, of course, wouldn't be here without their support throughout my career. This award is shared with my students and postdocs, past and present, who did the work that's recognized today. I'm really happy to share this with my family as well, who are here, some of whom are watching. Some are no longer here, but I know would be beaming with pride. I'm especially grateful for my wife, Lauren Babcock-Dunning, who's sitting in the back here with our 3 children, Isla, Marlo, and Calder. Thank you for your patience today throughout the ceremony. Thank you for your patience with me. And I'm just so happy to be here. And you've heard Calder give some remarks already during the presentation. So thank you to you all for entertaining us.
Okay, and now we move to the final award of the night, the Public Welfare Medal from the National Academy of Sciences. And I'm inviting Nancy up to the stage, your Home Secretary, to present this award. So, Nancy, take it away.
Thank you, Marcia. And first, congratulations to all of those who have just received their awards. It's my honor to introduce a scientist I admire tremendously, Dr. Mary Claire King. She's about to receive the National Academy of Sciences' most prestigious award, the Public Welfare Medal. First given in 1914 to recognize 2 men who were instrumental in the successful construction of the Panama Canal, the Public Welfare Medal honors exceptional individuals who advance science in the service of the public good. Mary Claire King has had a storied scientific career. A pioneer in human genetics, she is best known for proving that susceptibility to breast cancer can be inherited. Using clever strategies to enrich for a small percentage of patients with genetic predisposition, her group proved that individual mutated genes could greatly increase the risk of breast cancer. That was quite unexpected at the time. Her group narrowed down the chromosomal location that had to contain the culpable gene that she named BRCA1. That was paradigm-shifting work that led to much better survival for many women, but it is not why we are celebrating Mary Claire today. Instead, we're recognizing work that she's done to develop and apply genetic approaches to identify victims of violence and human rights abuses. Beginning at a time when it was far more difficult to do genetic analysis than it is today, Mary Claire used mitochondrial DNA sequencing to identify children abducted from their parents during Argentina's dirty war in the late 1970s and early 1980s. Her work led to the reunification of more than 130 families and to the creation of a national bank of genetic data in Argentina that will ensure more identifications can be made in the future. She extended her human rights work to identify victims in other countries and to identify the remains of soldiers missing in U.S. conflicts going back to World War II. Her application of genetics to find answers for families torn apart by repression and violence is a powerful example of how science can serve humanity. Mary Claire received her undergraduate degree in mathematics from Carleton College and her PhD in genetics from Berkeley. After her postdoctoral training at UCSF, she spent almost 2 decades on Berkeley's faculty before moving to the University of Washington, where she has been for 30 years as American Cancer Society Professor of Medical Genetics and Genome Sciences. In what you might call her day job, Mary Claire has contributed to our understanding of BRCA genes and genes responsible for many other disorders as diverse as hearing loss and neuropsychiatric disease. Mary Claire has been an NAS member since 2005 She is also a member of the American Academy of Arts and Sciences, the National Academy of Medicine, and the American Philosophical Society. She has received many, many major awards, including the Gruber Foundation Prize in Genetics, the Dan David Prize, the Shaw Prize, the Lasker Special Achievement Award for Medical Research, and the U.S. Medal of Science. It is a great pleasure to invite Mary Claire up to tell us her story. Take this off.
Thank you very much, Nancy and Marcia. This is the story of a band of grandmothers. It's an honor to tell it to you today. The story begins in Argentina in 1976, but to put it in context, I must tell you first 1 minute of Argentinian history. As you know, in World War II, Argentina, led by Juan Perón, was officially neutral, but Perón had been friendly with Axis dictators, and after the war, welcomed German and Italian fascists to Argentina. These men, and even more, many of their sons, became officers of the Argentinian military. In the 1960s and early 1970s, the Argentinian military and death squads carried out a chaotic internal conflict against leftist guerrilla groups. In Perón's last campaign in 1973, he was elected president by an overwhelming majority. But after his death, 2 years later, the military overthrew the weak successor government and began a program of suppression against their opponents. When the military overthrew the successor government to Perón's government in 1976, they began a program of suppression against their opponents. They dropped all pretense of legal processes, and this slide is an example of that. This is a slide from a training course for soldiers and police in 1976, and it indicates the subversives to be pursued by the military. The branches of the tree of subversion, shown here, include the leftist guerrilla groups, ERP and Montoneros, as one would expect, and also Christian Democrats, professors, journalists, liberal politicians, homosexuals, and so on. The roots of the tree are Marxism, Zionism, and Freemasonry. The military was effective. People disappeared. Among the disappeared were fathers and mothers of infants. However, generally no bodies of infants were found. The infants simply vanished. In 1977, the grandmothers of these vanished infants formed the Abuelas de Plaza de Mayo to demand information about their kidnapped children and grandchildren. Over the next years, the grandmothers accumulated a great deal of anecdotal evidence from midwives, housemaids, kindergarten teachers, about infants and small children who might have been kidnapped and given to military couples or their civilian collaborators. The grandmothers amassed hundreds of black binders of information. They searched for witnesses, organized data, followed suspicious soldiers, developed hypotheses, and marched. The grandmothers also asked for help from geneticists. Their question was this: if we locate a child whose identity is unknown, but we believe he or she is the child of disappeared parents and the grandchild of living grandparents, can we determine whether or not the child is in fact related to those grandparents? That is, can we extend paternity testing to grandparents if the parents are dead? It was not enough to prove that a child was not related to the people with whom the child was living. Rather, it was essential to know, in fact, who the child's biological family actually was. The grandmother spoke to the Human Rights Committee of the AAAS, and in early 1984, their request came to several of us. Led by Pierre Darlou of CNRS, it was straightforward to develop the statistics of grandpaternity testing based on Bayesian principles. That is, the probability of relationship given consistency of a genotype of a child with the genotypes of people who might be the child's grandparents. We developed a likelihood ratio that we called the Index of Grandpaternity, or Índice de Abuelidad. The grandmothers asked us to come to Buenos Aires to implement the index. My friends elected me as their representative. The timing was appropriate. In October 1983, the Argentinian military had been forced to call for elections after their disastrous invasion of the Falkland Islands. And in December 1983, human rights lawyer Raul Alfonsín was elected president. He established the Commission on the Disappearance of Persons, which invited me, along with 4 American forensic scientists, to help establish the number and identities of people disappeared during the dictatorship. I went to Buenos Aires in June 1984 for what I thought would be one trip. Before the visit, we had alerted the grandmothers that to implement the index, it would be necessary to identify highly informative genetic markers. Then, if it were possible to obtain a small blood sample from a child who could be one of their grandchildren, we could determine genotypes for each grandparent and for the child. In 1984, the most informative genes for this purpose were the HLA genes, which encode the histocompatibility antigens that must be matched for organ transplants. DNA markers were not yet used. Undoubtedly, HLA genes would be sufficiently informative to solve many cases. However, HLA typing was subject to serious practical limitations. Typing could only be performed on fresh blood and required multiple specific antigens as reagents. The grandmothers were not intimidated. They identified a laboratory in a hospital in Buenos Aires that was well equipped and competent in clinical HLA typing. When I arrived in Buenos Aires, the grandmothers took me to their office, a modest apartment in the city center. I opened the door. This poster covered the opposite wall. "Oh my God," I said, "where do we begin?" Rosa Rosenblit, the secretary of the Abuelas, looked at me. "We start with Paola," she said. Paola and her family were kidnapped in May 1978 from Montevideo. In 1983, based on good but circumstantial evidence, the grandmothers located a girl who had been registered for school by the then Deputy Police Commissioner Ruben Yavayen as his child. The grandmothers initiated legal proceedings to obtain genetic testing. Here is the pedigree. The question is whether Paola is a granddaughter of the Lugares and Greenspan families. The grandmothers had a court order to obtain blood from the child, and the surviving members of the Lugares and Greenspan families volunteered blood samples through the court. We extracted white blood cells and genotyped the HLA A and B loci. Mr. Greenspan had died after the disappearance of his daughter and granddaughter, but we were able to reconstruct his HLA types from his living children. Clearly, Paola could be a granddaughter of this family, inheriting HLA alleles from Mr. Lugares and Mr. Greenspan. We incorporated the population frequencies of all possible HLA haplotypes and also included blood groups, which were, of course, much less informative, leading to a grandpaternity index greater than 99.9%. The grandmothers presented this idea to the family court, petitioning for Paola's return. Yavayen and his wife, who had claimed that Paola was their biological daughter, were offered genetic testing by the court, but refused. The family court judge accepted the genetic data and ordered Paola's return to her family. Yavayen appealed the decision. He no longer claimed that Paola was his biological child, but instead claimed that he and his wife were the only family Paola knew, that they had rescued her, and that they treated her well. The appeals court, which still included judges appointed by the military regime, accepted Yavayen's argument and overturned the family court's decision. The grandmothers then took the case to the Argentinian Supreme Court, where they presented 5 arguments. First, it is not reasonable to ask whether a child's kidnapper, who was involved directly or indirectly in the torture and murder of her parents, is an adequate parent for his victim. Second, a child should not remain in the hands of kidnappers, regardless of the age of the child when kidnapped. Third, kidnapping is a crime. Is the situation different in Argentina because it happened on a large scale? 4, to abandon the search for kidnapped children in Argentina is to abandon a group of children who will not grow up in carefree innocence. As these children grow up, they will suspect the truth. What will be the effect of learning that they have been raised by the people involved, directly or indirectly, in the murder of their parents? And 5th, adults everywhere have a responsibility to children everywhere. Does failing to identify kidnapped children implicitly grant immunity to kidnappers? Does it give a sense of invulnerability to violators of human rights in other countries? The Supreme Court accepted these arguments. I was in Berkeley. Estela Carlotto, the president of the Abuelas, phoned me. In December 1984, Paola returned to her family. She walked through the door of the Lugares house, which she had not seen in 6 years, went to the bedroom that had been hers, and asked, "Where's my teddy bear?" Paula is a young teen, and now. The grandmothers had many other pending cases, and we immediately began working on them. For these first 5 families, for example, the genetics were straightforward, but working around the still powerful military to obtain compliance with court orders for restitution of children was always a struggle. A few months later, the case of Maria Jose Lavalle Lemos presented a new challenge for us. In 1977, Monica Lemos and Gustavo Lavalle, along with their young infant daughter, Maria, were kidnapped. Maria was returned to her grandmother, Heidi, a few days later. Monica was 8 months pregnant. Monica and Gustavo remained disappeared. In 1985, the grandmothers learned that a guard at the women's prison where Monica had been last seen alive had enrolled a girl in school with an obviously fraudulent birth certificate. Given the girl's age and the location of the prison, the grandmothers thought that the child might be Monica's daughter, born in captivity. The grandmothers were able to obtain a court order for a blood sample from this girl. We carried out HLA typing. We obtained a very high index of grandpaternity because grandmother Heidi, her undisputed granddaughter Maria, and the child who might be Maria Jose shared a very rare HLA haplotype. However, we knew that we were very lucky. Heidi was the only available grandparent. The chances were only 50-50 that Heidi would share an HLA haplotype with a granddaughter, let alone an extremely rare one. Clearly, in cases like this, we needed a better method. Besides, by 1985, we had many more cases. Many families were searching for their grandchildren. And for most families, we did not have information from all 4 grandparents. What to do? The grandmothers offered me a cup of mate and said, "Go back to Berkeley, Nina, and call us in a few weeks when you've figured it out." So I returned to Berkeley and went to see Alan Wilson, a father of molecular evolution and my PhD advisor 12 years earlier. Alan was using mitochondrial DNA to study human evolution, that is, ancient relationships between people of very different ancestries. It was immediately clear that mitochondrial DNA could also be used to identify people with much closer relationships. Mitochondrial DNA has 3 characteristics that make it perfect for these identifications. First, the mitochondrial DNA sequence of origin is highly variable genetically because it does not encode any genes and so has not been subject to selection. These 700 nucleotides are among the most variable in the genome. Second, mitochondrial DNA is inherited only from mothers. Both males and females, of course, have mitochondrial DNA, but the sequences are passed only through maternal gametes. Therefore, each person shares the mitochondrial DNA sequence of their sisters and brothers, their mother, her sisters and brothers, their maternal grandmother, and so on, the people in red on this slide. And because the sequence at the origin of replication is so variable from person to person, this sequence can distinguish one maternal lineage from another, based on any one maternal relative. And finally, mitochondrial DNA is haploid, so recombination is very rare, and the sequences are altered almost only by mutation. It was as if God had created mitochondrial DNA specifically for the grandmothers. But how could we obtain enough DNA to determine a sequence? DNA sequencing was very new and was still done by hand, one nucleotide at a time. For this problem, I called on Carrie Mullis, a friend from graduate school. Carrie was working at a biotechnology company and was developing the polymerase chain reaction, which allows, of course, many copies of DNA sequence to be made even from a single original molecule. Alan Wilson had just shown that the PCR product could be sequenced directly without needing to clone it first. This made PCR amplification and sequencing very quick, easy, and accurate. So I was able to call the grandmothers after about 3 weeks and tell them that it would indeed be possible to test many missing children, even with only one living maternal relative. Bueno, said Estella, when are you coming back? Back in Buenos Aires, to test the new approach, we amplified and sequenced the most informative region of mitochondrial DNA from Heidi and Maria Jose. The sequences were identical to each other and included sites that differed from all other sequences in the database that I had borrowed from the Wilson lab. The success of DNA sequencing for the Lavalle-Lemos family led us to recognize some stark realities. The number of families searching for grandchildren was rapidly increasing, now into the hundreds. Each family had circumstantial evidence and surviving relatives, but generally no one child for whom to test a hypothesis of identity. That is, instead of testing one child to one possible family, we now needed to test each child to many possible families. For this purpose, we needed a database of mitochondrial sequences from all the maternal lineages of families searching for children and from all the children who were coming to light. We needed genetic matching from one child to many possibilities through their genetic genealogy. Also, we needed proper security for chain of custody of samples and sequences. We needed all this housed in a voluntary and trustworthy national genetic data bank. Argentina created the world's first national genetic data bank in 1987, and it was put into use in service of kidnapped children and searching families. The immediate next question was, could we identify remains of deceased individuals by matching mitochondrial DNA sequences from their remains to sequences of surviving maternal relatives? In 1986, with the encouragement of the Commission on the Disappearance of Persons, a group of young Argentinian scientists had formed the Argentinian Forensic Anthropology Team to carefully exhume the mass graves uncovered at the end of the dictatorship. The team asked if we could determine the identity of victims by comparing mitochondrial DNA sequences from remains with the DNA of surviving maternal relatives. Until then, this had not been done. I decided to try teeth as a source of DNA. I reasoned that for an intact tooth, enamel would protect the pulp even after death, and we knew that the pulp of teeth is densely cellular. In principle, we could have also extracted DNA from bone, but the pulp of a tooth was less likely to be contaminated by handling. We treated the tooth like a diamond, splitting it and removing the pulp in a sterilized hood. In an undamaged tooth, the pulp proved to contain high molecular weight DNA. To develop the approach, we tested baby teeth from our friends versus blood from their mothers. We also tested DNA from teeth of a murder victim whom we were asked to study by colleagues in the New York State Police. Nothing to do with Argentina. We learned how informative mitochondrial DNA sequencing could be on its own, and when and how it could be combined with other information. The first case the Argentinian team asked us to address was the Manfield family. On the night of October 27, 1976, soldiers and police attacked an apartment in Avellaneda, an industrial city near Buenos Aires. Inside the apartment were Carlos Laudolino Manfield, his wife Angelica Zarate de Manfield, and three of their children, Carlitos, then 9 years old, Karina, then 4 years old, and Cristian, then 6 months old. Graciela, then 12 years old, was at her grandmother's house that night. Immediately after the attack began, Carlitos was shot in the forehead. He fell onto his sister Karina. The police fired into the children's bedroom, but Carlitos' body protected Karina, who was shot only in the leg. Cristian was not injured. His parents were killed. The bodies of the victims disappeared. Karina, Cristian, and Graciela were raised by their grandmother, Ana Ocampo. Karina grew up and joined the Argentinian Forensic Anthropology Team. In 1992, the team was working in Sector 134 of the Avellaneda Cemetery, a mass grave of those killed in that town by police and military personnel. The team excavated 324 skeletons from this grave, all identified as in or unknown. Almost all the remains were adults, but there was one skeleton of a child of approximately 10 years old with a bullet hole in the frontal bone of the skull. We were asked to compare DNA sequences from teeth of this skeleton and DNA from teeth of adult skeletons also in subsector D8, with blood sequences from living maternal relatives of the Manfield family. The results are on the slide. The mitochondrial DNA sequences of Karina, male child 49, and adult female 47 are identical. The mitochondrial DNA sequences of Ana Ocampo and adult male 46 are also identical. None of these sequences were identical to other remains from the mass grave, nor to any other mitochondrial DNA sequences in the Argentinian National Genetic Data Bank. The federal court accepted the anthropologic and genetic evidence, and the remains were returned to the family. This case, and the case that we worked on at the same time with the New York State Police, were the first cases of human remains identified through DNA sequences from relatives. We were soon asked by Chilhai the U.S. Armed Forces Central Identification Laboratory in Hawaii for help in identifying soldiers who were MIA from Southeast Asia, Korea, and World War II. Our first project was the identification of a journalist. In 1970, NBC journalist Wells Hangen went missing with 3 colleagues as he was reporting from Cambodia at the time of the invasion of Cambodia by American and South Vietnamese troops during the Vietnam War. We identified Mr. Hangen's remains by matching his mitochondrial DNA sequence to his sister. We identified dozens of other American MIAs, including Michael Blaise, who before this DNA sequencing had been buried in the tomb of the unknown soldier. We worked with the forensic anthropology team of Guatemala, who are proteges of the Argentinian forensic anthropology team, to identify remains from their mass graves. We worked in service of the International War Crimes Tribunal to investigate the murders near Srebrenica. We carried out some identifications in Rwanda, obviously only a token number. And with other groups, all working independently, we identified the Tsar and his family. We were very relieved when ABI sequencing was introduced and we could move on from sequencing one base pair at a time. During all of this work on the identification of remains, what was happening to the work of the grandmothers? As Rosa had predicted in the 1980s, as time had gone on, the grandchildren had come looking for them. In 2014, a young teacher named Ignacio learned that he might be among the kidnapped children and came forward. The Argentinian National Genetic Data Bank proved that he is Estela Carlotto's grandson. And in 2000, we identified Guillermo, the grandson of Rosa Rosenblit, a case that was profiled recently on 60 Minutes. By 2000, we had incorporated nuclear DNA analysis into the approach, so we had the specificity to determine relationships for cases of mitochondrial DNA sequences that are shared by multiple families, as in the case of the Ashkenazi Jewish sequence of Rosa and her grandchildren. Rosa is still alive at age 104. She told me a year ago that searching for the grandchildren kept her from growing old, and that when her grandson was found, when she was only 80, she knew that she must stay alive as long as possible to enjoy him. As of 2023, the grandmothers had found, or been found by, 133 grandchildren. As of this weekend, that number is 139. And genetic genealogy is useful worldwide. Now, of course, we use long-read whole genome DNA sequencing. I would like to end with the words of Argentinian, Chilean, American poet Ariel Dorfman. And finally, when the day comes, when they ask you to identify the body, and you see me, and a voice says, "We killed him. The poor bastard died. He's dead." When they tell you that I am completely, absolutely, definitively dead, don't believe them. Don't believe them. Don't believe them. Don't believe them. Thank you.